they are haploid. Because in meiosis I, what happens is you divide the two chromosome pairs, homologous chromosomes, one from the other, with one to one pole, the other to other, and they end up in new daughter cell right there. Okay, so these cells are haploid, right? The secondary spermatocytes, and here on down, all the cells are haploid. Okay? These cells then undergo meiosis II and produce daughter cells called spermatids, which are shown here, also haploid. In meiosis II, what you're basically doing is splitting double-stranded chromosomes into single-stranded. You're not changing the chromosome number, haploid to haploid here. Okay, the last phase of gametogenesis is right here. Spermatid to sperm cell. What kind of nuclear division here? What you call a trick question. No, there is no. That's why it's 1 for 1. What happens is what we call spermiogenesis. Went from the spermatid to the sperm cell. 3 major things. You see one of them right here, pretty common. Develop a flagellum, so the sperm is molded. Okay. What else?
Shed their cytoplasm.
Shed excess cytoplasm, which is not obvious here because these are not to scale. And what else? Third thing. Formation of aquisome, a little bag of digestive enzymes in the head of the sperm. So the sperm can literally digest its way through the protective barriers around the egg. Yes? Second one is shedding excess cytoplasm. So develop a flagellum, shedding excess cytoplasm, and formation of aquisome. Bag of digestive. It's like a lysosome, bag of digestive enzymes. Okay? And of course, that's not shown in here either. Right. And notice, one spermatogonium, how many gametes are there? 4. You want lots of sperm by the millions. Okay? All right, this is oogenesis. Where does oogenesis occur in the body of a female? In the ovary, but where in the ovary? Within the follicles. Each follicle, you've got a developing egg cell. That's where oogenesis is going on. In a follicle, in the ovary. Again, we start with a precursor diploid cell, oogonium. This divides its nucleus by mitosis, produces this cell, and another cell, another oogonium basically. And it's mitosis of diploid to diploid. Secondary, or the primary oocyte here, undergoes meiosis I and forms this and this. This cell, which is haploid, is the secondary oocyte, and the other cell is called what? A polar body. And again, this is not to scale. A polar body is tiny. A polar body is only a means of getting rid of excess chromosomes without wasting precious cytoplasm in the process. That's why cytokinesis, the division of cytoplasm, is unequal here. You want to keep most of the cytoplasm and the organelles associated with the egg, and not waste it on these polar bodies, which are going to degenerate anyway, even though they may divide. Okay, they're all going to degenerate. Okay? So this is the important cell, the secondary oocyte. This undergoes meiosis II. And when it completes meiosis II, it becomes an oval, a mature egg. But it's only going to do that until what happens, or if what happens. It's got to have a sperm cell penetrate the cytoplasm. That stimulates an egg cell to complete meiosis II, usually arrested in metaphase of meiosis II, and become a mature egg. And of course, chromosomes come together in the nucleus, now we've got a diploid cell again, 2 haploid gametes from an egg, and this is the zygote or fertilized egg. From now on, the division again is by mitosis. So diploid cells giving rise to diploid cells. And again, this is actually going to be in the oviduct, and as this cell passes down the oviduct and moves to the uterus, it's starting to develop. So early embryonic development occurs not in the uterus, but in the oviduct. And that embryo is en route to the uterine cavity. And by the time it comes out in the uterine cavity, it's not a fertilized egg anymore. What is it? What stage is it in? You know these stages. What's it called? It's called a blastocyst or blastula. So we look at this. This represents the fertilized egg, okay? That's the nucleus of the cell. There's the cytoplasm. That represents sperm. Actually, it's not quite accurate because the sperm usually, the head comes in and the tail is left on the outside. This may be general. The only important part is the head that contains the male chromosome. But notice how small the sperm is relative to the egg. So they said, most of the cytoplasm that makes up that diploid zygote from the egg. And that's where the organelles came from, for all of us. All our organelles are derivatives, okay, of our mother's egg cell organelles, not the sperm. So it was just a vehicle to get the male chromosome into the egg. And when I say shed excess cytoplasm, you can see there's not much cytoplasm in that sperm cell. It's a head with a nucleus, the male chromosomes, the acrosome, a midpiece which contains a lot of mitochondria to power the movement of the flagellum. That's all it is. You've got to get those chromosomes into the egg. That's the purpose of the sperm cell. Then it starts to divide by mitosis, 2-cell stage, 4-cell stage, 8-cell, or this only has 7, this one hasn't completed. But if you lose it, this is wrong. Only got 7, this hasn't finished, right? Then it goes to 16, 32, 64, 128, 256, 512, 1024, 2048. Keeps increasing by multiples of 2. Now it reaches a certain stage, it's about a 32-cell stage, solid ball of cells. What do we call that? That's the morula, which means berry. Sort of looks like a berry, right? Maybe a raspberry, blackberry, whatever. Okay?